Title: Genomic and Physiological Mechanisms Underlying Skin Plasticity During Water-to-Air Transition in an Amphibious Fish. Abstract: the terrestrial radiation of vertebrates required changes in skin that resolved the dual demands of maintaining a mechanical and a physiological barrier while also facilitating ion and gas transport. Using the amphibious killifish, Cryptolebius marmoratus, we found that transcriptional regulation of skin morphogenesis was quickly activated upon air exposure, 1 hour. Rapid regulation of cell-cell adhesion complexes and pathways that regulate stratum corneum formation was consistent with barrier function and mechanical reinforcement. Unique blood vessel architecture and regulation of angiogenesis was likely supported cutaneous respiration. Differences in ionoregulatory transcripts and ionocyte morphology were correlated with differences in salinity acclimation and resilience to air exposure. Evolutionary analyses reinforced the adaptive importance of these mechanisms. We conclude that rapid plasticity of barrier, respiratory, and ionoregulatory functions in skin evolved to support the amphibious lifestyle of K. marmoratus. Similar processes may have facilitated the terrestrial radiation of other contemporary and ancient fishes. Introduction. Devonian fishes seeded the first successful vertebrate invasion of land, where various phenotypic innovations promoted dramatic terrestrial radiation. What are the mechanistic changes that facilitated this pivotal adaptive leap to life on land? It is difficult to infer the physiological traits that enabled the original Devonian invasion of land. However, modern fishes have repeatedly evolved physiological and behavioral traits that enable exploitation of terrestrial niches. Extant amphibious fishes offer an opportunity to study the variety of mechanisms that may have facilitated the challenging transition of vertebrate lifestyles between aquatic and terrestrial habitats. The skin of amphibious fishes plays a critical role in maintaining homeostasis during air exposure. Histological studies have shown that a number of species have capillaries close to the epidermal surface, providing an effective surface for gas exchange. As well, ionocytes normally found in the gills are positioned close to the epidermal surface in some amphibious fishes and appear to be important in ion regulation. Cutaneous water transport, mucus production, and resistance to evaporative water loss likely also contribute to water balance during prolonged air exposure. In addition, the skin is typically a site of nitrogen excretion in fish out of water. But the skin must provide protection from pathogens and environmental hazards. Thus, the skin is multifunctional in amphibious fishes, but the mechanisms that resolve the opposing pressures of exchange versus barrier have received little attention. For the skin of amphibious fishes to become a crucial organ for oxygen transport, ion and water regulation, and nitrogenous waste excretion, and to provide a mechanical barrier out of water, we hypothesized that structural, physiological, and molecular changes must occur soon after exposure. We therefore predicted that a rapid and sustained regulation of molecular pathways associated with dermal, epidermal, structural remodeling, gas exchange, ammonia detoxification, and water and ion transport underpin the alterations in skin structure and function that unfold during this transition from immersion to emersion. To test these hypotheses, we used the amphibious mangrove rivulus, Cryptolebius marmoratus, which is a particularly opportune model system for studying the physiological and genomic mechanisms that support terrestrial acclimation. The species is native to the Western Atlantic mangrove forest from southern Florida to Brazil. They frequently escape water for terrestrial microhabitats, including the mud walls of crab burrows, damp leaf litter, or rotting logs for variable durations, up to 66 days. We compared two clonal strains of K. marmoratus during acclimation from water to air. One lineage is native to brackish water habitats in Honduras, HON, and the other to freshwater habitats in Belize, FW. We chose these lineages because of their different osmotic niches. Because their different osmotic niches may contribute to physiological differences in water and ion regulation, and because prior studies showed that the HON strain had a considerably higher resilience to air exposure relative to the freshwater strain. Lineages were independently perpetuated by self-fertilization, resulting in a purging of within-lineage genetic variation and creation of isogenic strains. Strains were acclimated to their native salinities, and then over the course of 1 week of air exposure, we measured changes in genome-wide gene expression, RNA-seq, skin structure, light microscopy, transmission, electron microscopy, and survival to infer skin mechanisms that resolve the multiple competing physiological demands of terrestrial acclimation. We predicted that genotype and osmotic acclimation would interact to provoke between-strain differences in the regulation of some of these mechanisms, and thereby provide additional insight into mechanisms that underlie variable terrestrial acclimation abilities. Finally, we used models of comparative protein evolution to identify genes evolving by directional selection in K. marmoratus, compared with other fish species that have limited or no terrestrial acclimation ability. We predicted that genes evolving by directional selection uniquely in the K. marmoratus lineage would be associated with the molecular pathways that regulate the species' unusual terrestrial acclimation abilities. Materials and methods. Animals. 2 isogenic hermaphroditic strains of K. marmoratus were maintained individually in the Hagen Aqualab at the University of Guelph, Ontario. A strain originally collected from Honduras, HON, in 1996 was held in the laboratory 15 part per thousand, 25 degrees Celsius for 20 years, around 60 generations. A freshwater strain, FW, captured in December 2012 at Long Key, Belize, in an open pool, 0.3 part per thousand, was held in the laboratory, 0.3 part per thousand, 25 degrees Celsius, for 3.5 years, around 10 generations. HON fish and those from the freshwater Belize site are genetically distinct. Fish were held singly in plastic containers in 60 mils of water under constant conditions, 12 hours, light and dark, 25 degrees Celsius. Reverse osmosis water and sea salt were used to formulate water to the appropriate salinity and changed weekly. Fish were fed live artemia 3 times a week. University of Guelph Animal Care Committee approved this project. Experimental design. HON and freshwater fish were air exposed for up to 7 days at 25 degrees Celsius. Before aerial transfer, tissues from immersed fish were sampled, control, and 0 hours. Upon air exposure, fish were maintained on moist filter paper, 15 part per thousand or 0.3 part per thousand, in plastic rearing containers, 99% humidity, and skin samples were collected at 1, 6, 24, 72, and 168 hours for transcriptomics. Fish were euthanized with tricaine methane sulfonate. Skin was dissected from the mid-region of the body by making a ventral incision and carefully removing extraneous tissue and muscle from the skin, and was then stored in RNA at minus 20 degrees Celsius. For histology, fish were acclimated to water or air, 0 hours and 168 hours, euthanized by immersion in ice-cold water, and then fixed and preserved for either collagen staining or transmission electron microscopy, TEM imaging. Survival. Survival was measured using a different set of fish that were air exposed for up to 7 days and monitored daily. The experiment was terminated if survival reached as low as 20%. Skin structure. We measured skin collagen content because it is a key contributor to the mechanical properties of skin, especially in amphibious species. And collagen is regulated in other tissues, gill arches and filaments, during air acclimation in K. marmoratus. For collagen staining, whole fish were fixed and processed as described previously. Tissues were then stained with picrocereus red, a collagen-specific dye, photographed under circularly polarized light, and the mean collagen hue for each picture was calculated. Samples for TEM observation were prepared using skin sampled from both HON and freshwater animals that were either immersion controls or had been exposed to air for 7 days. Samples were acquired from a standardized region of skin just posterior to the operculum and dorsal of the horizontal midline. Tissue was removed to include a small section of underlying epaxial muscle so as not to damage the skin and to provide orientation when sectioning. Isolated tissue was placed immediately into freshly prepared ice-cold glutaraldehyde fixative. Samples were fixed at 4 degrees Celsius overnight and then rinsed twice in sodium cacodolite buffer. Samples were post-fixed in osmium peroxide, rinsed twice in buffer, and dehydrated through a graded ethanol series, then immersed in propylene oxide and transitioned into quetal spur resin. Quetal spur resin, followed by, final infiltration into quetal spur resin was carried out overnight, after which sample pieces were placed in embedding molds and polymerized at 65 degrees Celsius overnight. Sections were prepared using a Lycra ultramicrotome stained with uranyl acetate and lead citrate and viewed in an FEI Techni 20 transmission electron microscope. Skin morphometric analysis was conducted using ImageJ software. Subapical desmosome 2 was the second desmosome observed in the subapical region of the epidermis, and its depth was determined by tracing its length. On average, for each fish, the lengths of typically 6 to 10 desmosomes were measured and then averaged to give an N of 1 for that animal. A total of 158 desmosomes were measured for 20 animals. Skin transcriptome. Transcriptomics data collection and analyses were described in Blanchard et al. 2019. Briefly, tissues were homogenized and RNA was extracted using RNA-Z purification kits. Individually indexed RNA-seq libraries were prepared using NAB-Next RNA library preparation kits for Illumina. Libraries were pooled, and sequence data were collected across 4 lanes of Illumina 4000. 5 biological replicates were included per treatment group, except for 2 treatments, Hans strain 72-hour air exposure and Hans strain immersion control, that had 4 replicates. Sequence reads were mapped to the K. marmoratus gene set reported in Wright et al. 2017 using STAR, and read counts were generated using HT-seq. We retained genes that had greater than 10 read counts for at least 4 of 5 biological replicates within any treatment group. Read counts were log2 transformed and normalized for gene length and total library size in EDGE-R. Differential expression analysis was performed in Limma, where strain, on and freshwater, and time, 0, 1, 6, 24, 72, and 168 hours, were specified as main effects, and strain by time as an interaction term, including false discovery rate adjustment of p-values. RNA-seq read sequences are archived at NCBI. A matrix of per gene per sample raw read counts and R scripts that detail all these analyses are available in a GitHub repository. A list of all genes included in the analysis, including annotation, average expression levels, and p-values associated with the results from statistical tests, is in Appendix S1, available from Figshare. Detecting positive selection. Positively selective molecular changes that are lineage-specific underlie adaptive phenotypes that may be unique to a species. They leave recognizable footprints in the genome, for example, in the amino acid sequence of proteins. Such footprints are detected when sequences are compared between orthologous loci among relatives. We used OrthoMCL, which is a program that filters orthologous proteins and guidance to remove unreliable coding sequence alignments and alignment positions. We then used phylogenetic analysis by maximum likelihood to identify positively selected sites on targeted lineages, in our case, those that are specific for K. marmoratus. Because K. marmoratus is the only amphibious species among those included in this analysis, the genetic changes that support their unique amphibious lifestyle are likely among those showing positive selection specifically in the K. marmoratus lineage. We downloaded protein and coding sequences in FASTA format of genome assemblies from K. marmoratus, Astrofundulus limnaeus, Notobranchius fusari, Massilia formosa, Fundulus heteroclitus, Oryzius latipes, Gastrosdius aculeatus, Tachyfugu rubripes, and Daniorerio from NCBI and the Broad Institute. We chose these species because they had well-assembled and annotated genomes and are all strictly aquatic, not amphibious species, and they represented a range of phylogenetic distances from K. marmoratus. To assign a database of orthologous sequences from these 9 sets of gene-protein FASTA sequences, we used OrthoMCL, and the following criteria were applied to achieve optimal clustering resolutions. Similarity p-value less than 1 times 10 to the minus 5, protein percent identity less than or greater than 40%, and MCL inflation of 1.5. In a preliminary clustering step, compliant FASTA files were established by applying the module OrthoMCL adjust FASTA. The OrthoMCL module OrthoMCL filter FASTA was used to remove proteins of poor quality to finally generate the good protein FASTA file, compiling all proteins with specific assigned headers for the subsequent all versus all blast. To build orthologous groups, we conducted an all versus all NCBI blast. With the OrthoMCL module, OrthoMCL blast parser, similarities were then summarized within a file that was loaded to MySQL by the OrthoMCL load blast module. the ortho-MCL pairs and ortho-MCL dump pairs files modules, then found, sorted, and created results files of all protein pairs. The MCL and ortho-MCL to groups modules generated the groups.txt file. We then used a custom Python script to extract only unique genes that were represented, 1, in all species, and 2, owing to their critical phylogenetic distance and poorly sequenced genomes, in all species except Danielrerio and or Takafubu rubripes and or Gastricius aculeatus. We then extracted all corresponding nucleotide sequences from the coding sequence FASTA file by applying another custom Python script. Guidance 2 was then used to remove unreliably aligned positions in the multiple sequence alignments before testing for positively selective sites with PAML4. We conducted the analysis twice using different aligners, 1, muscle, and 2, MAFFT. Figure S15 lists all guide trees that were used to fulfill branch site tests for positive selection in the K. marmoratus lineage using PAML. We inferred positive selection in K. marmoratus when the alpha value for the likelihood ratio test was less than 0.01. Statistical analysis. Treatment effects on collagen staining were tested using two-way ANOVA with time and strain specified as main effects, including an interaction term, followed by a post-hoc Holmes-Sidak test. Treatment effects on desmosome length were tested using two-way ANOVA with time and strain specified as main effects, including an interaction term. Differential expression analysis was performed in lemma, where strain, HON and freshwater, and time, 0, 1, 6, 24, 72, and 168 hours, were specified as fixed main effects, and strain by time as an interaction term, including false discovery rate adjustment of p-values. We examined 3 sets of transcriptional responses. 1, transcripts with a significant temporal effect that was similar between HON and freshwater fish, a shared air exposure response gene set. 2, transcripts with a significant time by strain interaction, genes that differed in their air exposure response between HON and freshwater fish. And 3, transcripts that were not responsive to air exposure, but were constitutively differentially expressed between HON and freshwater fish, strain genes. Shared air exposure response genes were grouped by temporal patterns of co-expression, which were defined by the time post-air exposure at which expression was first significantly different from immersion controls. This grouping was performed separately for upregulated and downregulated compared to immersion control genes. For functional inference, clusters of co-expressed genes were examined for gene ontology, GEO, and KEGG pathway enrichment using DAVID. Results and discussion. Overview. We found remarkably rapid molecular changes in the skin within 1 hour of air exposure. Genes associated with skin remodeling, including those involved in skin morphogenesis, cell junctions, and extracellular matrix, were quickly upregulated. Histological images of the skin verified that after 7 days in air, junctional complexes and collagen were altered. Collagen content were altered. We also found a rapid upregulation of aquaporin genes and those genes involved in paracellular permeability, underlying the importance of skin osmoregulation. Large ionocytes with elaborate crypts were observed in the skin of air-exposed fish that were uniquely associated with basolateral capillaries. We also showed that genes linked to angiogenesis were quickly upregulated, consistent with an extensive capillary network close to the skin surface. Finally, our analyses of gene loci evolving by positive selection in K. marmoratus identified a number of genes involved in skin morphology, morphogenesis, and angiogenesis, reinforcing the critical importance of the skin for transitioning effectively between water and air. Skin morphology. Air exposure induced rapid but mostly transient changes in the transcription of genes associated with skin remodeling, figure 1. Transcription factors were enriched among the genes upregulated by 1 hour in air, including those known to regulate skin morphogenesis, such as transcription factors P65 retinoic acid receptor, Kruppel-like factor 4, and transcription factors AP1, like Jun and Foss. Amphibian skin remodeling during metamorphosis, is thyroid hormone dependent. We observed parallel upregulation of THRA and RARG within 1 hour of air exposure, which is consistent with a convergent role of thyroid hormone signaling in skin remodeling during terrestrial transitions between amphibians and amphibious fish. This rapid and transient burst in gene expression that we observed is likely important for initiating skin remodeling, which is important for resilience to the aerial environment. At cell junctions, multiple structures serve to attach epithelial cells together in sheets and to the basement membrane and extracellular matrix, ECM, and regulate the exchange of small molecules and the passage of water. We found a rapid upregulation of genes involved in desmosomes and adherens junctions that provide strength to tissues, figure 1a, table S2. Focal adhesions are integrin-containing complexes that attach epidermal cells to the basement membrane and ECM. Here we also observed peak increases in the KEGG pathway focal adhesion 6 hours after air exposure, including transcripts for integrin subunits and ligands, figure 1a, figures S3, S4. Furthermore, gap junctions, for example, connexin-containing complexes, provide channels through which small molecules may pass between adjacent cells and tight junction complexes, for example, claudins and cludins. And tight junction proteins regulate paracellular permeability to water and small molecules. The molecular components of these structures and the pathways that regulate them were all transcriptionally regulated within hours of exposure, figure 1a, figures S5 and S6. Furthermore, TEM observations confirmed that differences in the configuration of specialized epidermal structures associated with the functional integrity of the epidermis occurred following air exposure, figure 2. Desmosomes serve as the intracellular glue that is required for epithelial integrity, and desmosome proteins are evolving by directional selection in K. marmoratus, see below. We conclude that connections between keratinocytes that interface with the environment are, at least in part, regulated through desmosomes, and that this is likely to be adaptively important for mechanical reinforcement of the skin during air exposure. Structural constituents of the extracellular matrix were regulated during aerial acclimation. The gene ontology terms collagen and extracellular matrix organization were enriched among transcripts downregulated between 1 and 7 days in air, figure 1a, table S6, including the downregulation of at least 23 collagen transcripts. This suggests that reduced collagen synthesis is an important component of skin remodeling during air exposure, which is consistent with our observed reduction in collagen staining in skin sections by 7 days in air, figure 3a. The thickness of the collagen layer, or collagen content, may affect exchange across the skin, but this is unknown, and the diffusion distance for gases, water, and ions will be set, more importantly, by the closeness of the capillaries to the surface. Collagen is also critical for mechanical durability of the skin, and less collagen during terrestrial sojourns may be advantageous for locomotion or other functions. But further studies are required to understand the role of collagen in K. marmoratus skin. TEM observations indicate that collagen is prominent in at least 4 regions. 1, the stratum compactum in the lower reaches of the dermis. 2, the scales. 3, the dermis-epidermis interface. And 4, the epidermal and subepidermal vasculature, figure 3b to E. Collagen in the subepidermal region of K. marmoratus skin exhibited an unusual structural arrangement relative to that reported for other fishes, as it appears in loosely ordered bundles, compared with the highly ordered and deep plywood-like lamellae localized to the upper reaches of the dermis in other fishes. In amniotes, the stratum corneum, SC, forms the outermost layer of the epidermis and functions as a barrier to protect from dehydration, crucial for the terrestrial radiation of the amniotes. Though fishes lack many of the key proteins that constitute the mature stratum corneum in amniotes, our transcriptional data provide evidence that the molecular machinery that supports the formation of the stratum corneum in amniotes is activated during air exposure. The transglutaminases, for example, TGM1 and TGM5, that govern the process of protein cross-linking that produces the stratum corneum have been found in fish. In K. marmorata skin, TGM1 and TGM5 were significantly upregulated within 1 hour of exposure, figure S8. The other key proteins that regulate this pathway were also upregulated after 1 and 6 hours in air, for example, EVPL, PPL, KAZN, figure 8. Furthermore, the sphingolipid metabolism pathway, which produces lipids integral to barrier function of the stratum corneum in amniotes, is upregulated between 1 and 7 days after air exposure. TEM images provide no evidence for the formation of a stratum corneum, so the physiological manifestations of these stratum corneum-associated molecular responses merit further inquiry. Water ion transport. Cryptolebius marmoratus are able to effectively counteract desiccation while out of water. After several days in air, they increase water influx across the skin, whereas water efflux does not change, and fish maintain whole body water levels. Enhanced water uptake may involve aquaporins or changes to paracellular tight junctions. And upon air exposure, we observed rapid upregulation of aquaporin 3, as well as claudin 10 and occludin tight junction transcripts. In air-exposed fish, TEM revealed 2 types of subapical tight junctions of the epidermis. The most common were deep tight junctions, around 200 nanometers, which link most cells in the epidermis. A second type of tight junction observed was a shallow or leaky tight junction, which linked ionocytes and adjacent interdigitating cells in an arrangement that was similar to the relationship between seawater fish gill ionocytes and interdigitating accessory cells. Shallow tight junctions directly interfaced with the environment within ionocyte apical crypts. They were frequently observed in TEM images from pond fish acclimated to air, but none were observed in freshwater fish, despite the unusual presence of apical crypts in these animals, not a feature of freshwater gill architecture. In the seawater fish gill epithelium, ionocyte apical crypts established a microenvironment within which paracellular movement of sodium across leaky junctions can occur, down an electrochemical gradient established by transcellular chloride secretion through the chloride channel CFTR. Therefore, the enigmatic presence of deep apical crypts without leaky junctions in ionocytes of air-exposed freshwater fish most likely reflects the need to create a microenvironment for solute transport, not for sodium secretion, but possibly for ammonia trapping. In line with these observations, pond and freshwater animals differed in their air-induced expression of key components of fish ionocytes, including sodium-potassium ATPase, CFTR, and claudins. Claudin 10 proteins are proposed to selectively facilitate sodium movement, and here we observed much higher mRNA levels in pond fish than in freshwater fish. Therefore, our observations suggest that sodium regulation through leaky tight junctions that reside within apical crypts of Hahn epithelial ionocytes is important for aerial acclimation in this strain. Ammonia trapping rhesus RH proteins are localized to the apical crypt of epidermal ionocytes in K. marmoratus and are thought to facilitate cutaneous ammonia excretion on land. The genes coding for 3 RH isoforms, RH types A, B, and C, and V-type proton ATPase subunits were upregulated after 6 hours of air exposure, consistent with the acid trapping model proposed for branchial ammonia excretion. Also, glutamine synthetase, which detoxifies ammonia, is significantly increased at 1 hour following air exposure. Interestingly, gene expression of the sodium proton exchanger NAG2, also involved in ammonia excretion, was 50-fold higher in Hahnfish compared to freshwater fish. This difference may reflect different strategies for ion transport and possibly ammonia excretion between fish acclimated or adapted to different salinities. Respiration. Morphological hallmarks of cutaneous respiration in vertebrates are augmented vascularization of the skin and the presence of blood vessels in the upper reaches of the epidermis, which reduces the blood-to-water diffusion distance and enhances epithelial gas transport in air. Our TEM images show that by 7 days in air, the diffusion distance from air to blood in epithelial capillaries is as short as 0.23 microns. To our knowledge, this is the shortest cutaneous diffusion distance yet measured in a fish. By comparison, diffusion distance across the skin of other amphibious fishes, as well as the gill epithelium of fishes in general, is at least 1 order of magnitude greater. TEM observations also revealed the presence of blood vessels at the base of ionocytes, establishing a blood-to-water bridge across which solutes could be efficiently transported. Cutaneous angiogenesis and key angiogenesis regulating genes were elevated within 1 day of air exposure in K. marmoratus, as reported in our companion study. Here, we show that genes involved in the KEGG pathway VEGF signaling were enriched among the genes significantly upregulated by 6 hours in air. And angiogenesis and vasculogenesis are biofunctions that were significantly enriched among the genes upregulated by 24 hours in air. These include transcription factors, XBP1, NR4A1, and SRF, which regulate VEGF-dependent angiogenesis. Furthermore, bradykinin receptor promotes angiogenesis through nitric oxide signaling, and we found parallel upregulation of BDK-RB2 transcripts and NOS1 by 6 hours in air. We conclude that angiogenesis, coupled with blood vessels in very close proximity to the apical surface and at the base of ionocytes, is important for shifting respiratory and ionoregulatory functions from the gill to the skin during aerial acclimation. Different responses between HON and freshwater fish. Considering 7-day survival data, the HON fish were more resilient to air exposure than freshwater fish. This could be due to genetic differences between strains or to differences in acclimation environment. Though the freshwater strain inhabits a very different native habitat, freshwater, than HON strain animals, brackish marine, likely resulting in evolved differences in physiology, the experiments reported here were not designed to distinguish genetic, heritable, from acclimation differences. Variable resilience could be related in part to skin function. Among the skin genes that were differentially expressed between strains during aerial acclimation, strain by time interaction, we observed 3 clusters of co-expressed genes. Set 1, figure 1b, is significantly enriched for the geoterm cell cycle. These genes show a transient spike in expression at 1 hour for HON fish, but gradual downregulation and then recovery in freshwater fish. Set 2 is enriched for the geoterms proteasome and lysosome. These genes show gradual upregulation only in freshwater fish, while little temporal response in HON fish. Set 3 is enriched for the geoterms translational initiation, cell junction, and sodium transport. These genes show gradual downregulation only in freshwater fish, with little temporal response in HON fish. Immediate differences in cell cycle regulatory response may indicate differences between HON and freshwater animals in their ability to sense and respond to cellular stress incurred upon air exposure, set 1. Cellular damage may have accumulated in the less resilient freshwater strain, such that proteolytic activity is upregulated primarily in that strain, set 2. HON and freshwater animals differ in their regulation of structural and functional components of the osmoregulatory apparatus in ionocytes, set 3. Indeed, in addition to the sodium transport genes, including in sodium potassium ATPase, sodium bicarbonate transporter, and sodium proton exchanger, a number of other key gill osmoregulation genes are differentially expressed between strains, including the V-type proton ATPase, CFTR, carbonic anhydrase, claudins, and aquaporin. We conclude that several differences in the regulation of skin pathways from acclimation and/or adaptation to fresh water may constrain the ability of K. marmoratus to transition between water and air. Considering that Devonian tetrapods were likely derived from marine or brackish lineages, and that brackish and terrestrial environments share challenges posed by dehydration, perhaps salty aquatic environments favor osmoregulatory physiologies that are more easily co-opted for terrestriality than freshwater environments. Our data are consistent with this hypothesis and suggest that physiological differences in ionoregulation are mechanistically associated with terrestrial acclimation abilities. Protein evolution. Our initial test for positive selection in K. marmoratus protein sequences included 1,073 single copy orthologous gene loci in all 9 fish species included, and revealed 24 genes showing positive selection in the K. marmoratus lineage only. An extended analysis expanded the query set to include orthologs present in all species except one of either Daniorerio, Takifugu rubriferis, or Gastrocilus aculeatus. This included 461 further genes for analysis and revealed 16 more gene loci evolving by positive selection in the K. marmoratus lineage. Because of the constraints for confidently identifying single copy orthologs across multiple taxa, this analysis queried only around 4% of the genes in the K. marmoratus genome. We detected positive selection in genes involved in skin morphology and morphogenesis, including 2 key genes that contribute to desmosome structure, desmogliin and plakophilin, and in transcription factor Kruppel-like factor 9, KLF9, which regulates skin morphogenesis. Desmosomes are an evolutionary innovation of vertebrates and share a conserved role in maintaining epidermal integrity to mechanical stress, including in amphibians. Desmocolin and desmogliin genes have expanded in the tetrapods, and desmosome genes are subject to selection during reinvasion of aquatic environments in mammals, consistent with a role for desmosomes in terrestrial to aquatic adaptations. Positive selection was also detected in heart and neural crest derivatives expressed protein 1, HAND1, which is required for angiogenesis and regulates the expression of many angiogenic pathways, including VEGF, angiopoietin, and ephrin signaling. Pathways related to skin remodeling, desmosome function, and angiogenesis are also among those showing patterns of differential regulation during terrestrial acclimation. We conclude that some of the proteins evolving by positive selection within the K. marmoratus lineage are adaptive for supporting skin plasticity, which enables the amphibious lifestyle of this species. Conclusions. Amphibious abilities have evolved 87 times independently among the extant fishes. Many species of amphibians transition between aquatic and terrestrial forms during metamorphosis, and 3 lineages of terrestrial mammals have transitioned back to fully aquatic forms, cetaceans, pinnipeds, and sirenians. This diversity provides ample opportunity to study the conserved and divergent mechanisms that support transitions between aquatic and terrestrial physiologies that are so important in the history of vertebrate diversification. So far, few studies have detailed the mechanisms that underlie skin plasticity and evolution during air exposure. Our integrated molecular, structural, and evolutionary analyses revealed the importance of rapid regulation of cell-cell adhesion structures, molecular pathways that govern skin morphogenesis, and evolution of desmosome proteins to mechanically reinforce and buttress barrier functions of this tissue. The challenge of supporting respiratory and ion exchange functions is associated with ionocyte structures and rapid regulation of ionoregulatory pathways, angiogenesis, evolution of the angiogenesis regulating proteins, and an unusual blood vessel architecture that links very short air-to-blood diffusion distance with delivery to energetically demanding ionocytes. Studies in marine mammals and amphibious gobies provide evidence of parallel importance of skin morphogenesis and ion transport mechanisms, respectively, in aquatic to terrestrial transitions. Overall, our data reveal that when K. marmoratus transitions from water to air, the skin quickly alters its barrier and exchange functions to delicately balance these dual purposes on land. Now I'm going to take a few minutes to walk you through some of the figures. Figure 1 is functional enrichment of co-expressed sets of genes. So we've got 3 different panels here, 3 different sets of heat maps. And in the heat map, the colors, each row is a different gene. Each column is average expression for a treatment. And the color of a cell indicates whether a gene is upregulated, which is yellow, or downregulated, which is blue, relative to the 0 hour immersed in water treatment. Okay. Each column is time, consecutive time points out of water in air. Okay. And one panel here is for the freshwater strain, and another panel here is for the Honduras strain. Okay. We've got 3 sets of genes here, set A, B, and C. Okay. So the set that we're highlighting in panel A are transcriptional responses through time, so a significant time effect, but no difference between the 2 strains. So basically a conserved transcriptional response, no matter which strain, to aerial exposure. Okay. Panel B are genes that show a time effect that differs between the 2 strains. So a strain by time interaction. Okay. And we've got basically 3 different patterns here. Set 1 are genes that basically drop quickly in expression and then recover back up in the freshwater strain, but show not much of a transcriptional response in the Honduras strain. Set 2 is a group of genes that show gradual increase in expression through time in the freshwater strain, but no change in expression through time in the Honduras strain. And set 3 is a set of genes that show a decrease in expression through time in the freshwater strain, but no response through time in the Honduras strain. And panel C are genes that don't show a response through time, don't really show an acclimation response, but that just show constitutive differences in gene expression between the 2 strains, no matter what time. So a main effect of strain only. Okay, so we got a set of genes here in the top half that's more highly expressed in freshwater than Honduras, and the reciprocal pattern below. And what we have in the word bubbles here are the gene ontology terms that are enriched within each cluster. Okay, so for panel A, genes are organized by when they're first transcriptionally different from the 0 hour exposure. So we basically got 3 waves of exposure, a set coming on early, a set coming on still pretty early, within 6 hours, 24 hours, and then sets that come on a little bit later. And then here are genes that decrease in expression within 1 hour, decrease in expression at 6 hours, and so on, that take longer. Okay. So for each of these clusters of co-expressed genes, we test whether there are gene ontology terms for those genes that are enriched within that set relative to the whole data set. Okay. And the basically the size of the enrichment, or the fold enrichment for that geo term, if it's statistically significantly enriched, is scaled by font size. Okay. So for example, for this set here, for genes upregulated by 1 hour exposure, transcription factor activity, which this little teeny set here, is enriched only 2.3 fold, significantly enriched, but statistically significantly enriched, but only a fold enrichment of 2.3, whereas desmosome is enriched 12.3 fold, so much larger font. Okay. And so we can see for this set of genes that are spiking at 1 hour after immersion, we see a whole pile of genes showed associated with the desmosome coming, transcriptionally upregulated responses to cytokines of transcription, pilot transcription factors coming on here as well. And then here we've got integrin complex that's really sort of coming online, and then genes involved in regulation of intracellular pH in the sphingolipid metabolism pathway coming online by about 1 day. Okay. And then we see really profound downregulation of genes associated with collagen and the extracellular matrix. What we see is a massive enrichment of genes associated with the MCM complex in this time by strain interaction set. Okay, these are genes that are really important in regulating the cell cycle. And then downregulated in freshwater, but no regulation in Honduras is proteasome complex, and upregulated in Han, not regulated in, start that over again. Set 2 are genes that are upregulated through time in freshwater, not regulated through time in Han. And then set 3 are genes that are downregulated in freshwater, not regulated in Han, again, associated with translation initiation and a few other things. Okay, so hopefully that gives you some sense of what's going on with figure 1 here. This is just sort of giving the big picture of transcriptionally what's going on under the hood for responses that are shared between the strains and that differ between the strains. Let's look at figure 2. This is basically looking at responses associated with a desmosome during aerial acclimation. Okay, so panel here is just a cartoon of the desmosome structure, looking at the proteins that constitute the desmosome. In panel B here, we're looking at a structural physiological response to aerial acclimation that's associated with the lengthening of desmosome. Okay, so the length of the desmosome increases by 7 days in air, those the open bars, relative to the immersion controls, which are the black bars. Okay, so whether you're the freshwater strain or the Honduras strain, as you go from water to air, the length of the desmosome increases. Okay, and we can see that visually in these representative electron microscopy images. These are shown for C and D are for freshwater fish, the freshwater strain. E and F are for the Hon strain. C and E are at 0 hour. D and F are at 168 hours. Okay, so C to D is freshwater fish immersion to D immersion at 7 days. Okay, and then the same thing for Hon strain. Hon is E immersed in water, and then after 7 days in air in F. Okay, and you can see the length of the desmosome is getting longer, right? And these arrows are pointing to, I think, desmosome 1 and desmosome 2. Okay, yeah, those are the black and white arrowheads. Then what we see in these remaining line graphs are the log2 expression levels for a bunch of the genes whose proteins are involved in the structure of the desmosome, as we can see up here in panel A. So you see desmoplakin, G, desmogliin 2 and H, desmocolin 2 and I, and plakophilin 3 and J. Okay, and we can see their temporal transcriptional response. Remember, 0 hours is in water, and then 1 hour out of water in air to 7 days out of air in air for both strains. Okay, the Honduras strain is the black, and the freshwater strain is the gray. You can see that all these really key proteins that make up a desmosome are strongly and rapidly upregulated upon immersion, upon exposure to air. Okay, figure 3. This is looking at skin collagen and skin structure. So we notice that skin collagen staining indicates lower levels of collagen in freshwater fish, which are these gray boxes, compared to the Hon fish, generally speaking. And the treatment affects it as well, right? So a decrease in abundance of collagen as you go from immersion day 0 to immersion day 7. Right, and we see this is, decrease in the transcription of collagen genes is really strongly downregulated over time with immersion that we saw in the very first figure. Okay, these TEM images, the electron microscopy images over here, show a really conspicuous collagen abundance in 4 different regions of the skin, including in the stratum compactum here in panel B, in the scales here in panel C, in the base of the epidermis and the upper reaches of the dermis and the collagen fibers that are really loosely dispersed here. And then in E, collagen fibers are associated with these integumentary blood vessels. Okay, so here CF is collagen fibers in panel B. SC is the stratum compactum and is nuclei. Okay, panel C here, this is a scale, SCL. And again, the CF are collagen fibers that are associated down there. Panel D, again, this is the dermis to epidermis region. Okay, and we see the collagen fibers associated down here. Okay, and in panel E, we see, capillaries, again, super close to the skin surface. We've got these micro ridges up here. We've got some of these filament-containing cells over here. N is for nuclei. C, again, is for these collagen bundles that are associated with the base of these capillaries. All right. Figure 4 are transmission electron microscopy images of epidermal ionocytes in the air-exposed fish and C of the skin blood vessels. Okay, so what we see here are these really deep apical crypts, these AC in air-exposed ionocytes in panel A, the freshwater fish, and panel B, the Hahn fish. And in both the freshwater fish and the Hahn fish, a dense intracellular tubular networks indicated by these black arrows, okay? And deep, tight junctions are the black arrow heads in A. They link ionocytes to the adjacent epidermal cells, okay? And in a B here, we've got this blood capillary residing right at the base of an ionocyte, a mitochondrial-rich cell, an MRC, okay? And in panel C here, we see these blood capillaries that are really extremely close to the apical surface of the epidermis. Again, less than a quarter micron, resulting in a really, really short diffusion distance, okay? So we see this in the scale bars here, okay. And those are the figures. I hope you enjoyed this narration of the manuscript.